All right, guys. So, hello. So I decided to try something new today. Um, and just record like the lecture for today or what we're learning for today. So today we are learning about DNA. So what we're gonna go over are what is DNA? Um, what are the nitrogenous bases of DNA? Nucleotides, we'll learn about nucleotides, maybe seem a little bit familiar, tying back to our biomolecule unit. And how does DNA look like? So what is the structure of DNA? How is how does it basically work well together? So let's start out by what is DNA? Well, DNA stands for deoxyribonucleic acid. So it's the blueprint of life. And by definition, our DNA is basically a hereditary molecule. From now on, I'll be saying like these vocab terms, hereditary, traits, um, things like that. And all this means is that it stores information about our traits, our proteins, and just instructions on how to make our bodies, right? So how to make organisms. Um, so let's go back to basically the beginning here. So here I have a couple of images. So everything that is under basically circled here in the red are all eukaryotics. So eukaryotes. And then after that, I have my bacteria here, which is a prokaryote. And then lastly, over here in the green is a virus. So all of these organisms plus our virus they all have DNA or some sort of genetic information, whether it's DNA or something called RNA, in which we will learn when we get back from break. Um, so all of these organisms have DNA. Virus is something that's kind of special in that viruses are not considered not considered living, but rather they are considered non-living just because um, they do not have organelles. Or they can't metabolize. So they can't do respiration, they can't do photosynthesis, they need a host. But we'll come back to viruses later on. So going back to DNA, so let's look at it at the molecular level. So DNA is a polymer of a nucleic acid. So remember when I talked about our biomolecules, so things like lipid, carbohydrate, proteins, and nucleic acid. Well, specifically, we are going to be focusing on nucleic acid for our next unit. So the monomer of basically a nucleic acid of the DNA is called a nucleotide. So what is a nucleotide? Well, our nucleotide is made up of something called a sugar, a phosphate, and a base. So these three parts, again, make up our nucleotide, a sugar, a phosphate, and a base. They're very, very essential and very important to how our DNA is basically structured. So remember that it's the monomer or the building block of DNA. So when we look at DNA, we can see here, especially in this picture, to my right, to your right. So DNA is structured basically in two long strands. So our nucleotides, the monomer for our DNA, are arranged in two long strands that form a spiral. So that's why it kind of like spirals around, right? Like a little spring. So they spiraled around called we call it a double helix. So this means that our DNA is double stranded. So we have two strands of basically a bunch of nucleotides that are stuck together um, that form together along basically the side of each DNA. So if we look at our DNA and we look at the nucleotides for each side, so nucleotides run anti-parallel to each other. So when we look at the way it's structured, see how this side on the left side of the DNA molecule here, I have my phosphate and my sugar here, and they're all basically running upright, right? So they're running upright. And then on this side, they're running the opposite way. So they're running the other way. So basically like if they're flipped over. This makes it anti-parallel to each other. So to get a little bit more into depth is that 5' to 3' has to do with the oxygen in our nucleotide. So going back to the last slide, our nucleotide starts from the phosphate group. So the oxygen here that's available to attach to another phosphate group, and the oxygen here. So this has to do with the way the oxygens are positioned. So that's why we biologists and chemists and just scientists in general talk about 5' to 3', the way that these oxygens are positioned. So seeing how it's phosphate connected to a sugar, to another phosphate, sugar, phosphate, sugar, phosphate, sugar, it has to do with the way that it's connected through each oxygen. So it's from 5' to 3'. So they run anti-parallel to each other. So parallel is side by side and they run anti-parallel to each other. So it's flipped. All right, so moving on. So let's look a little bit more into depth about our DNA. So we talked about nucleotides and how they basically run anti-parallel to each other and they make up the double strands here. So as you can see, the double strands here are the sugar phosphate backbone. So we're just talking about the sugar phosphate backbone, but now let's look at what these things in the middle are called, our nitrogenous bases. So this is where essentially where we store our genes, the genetic material. So genes are stored in the DNA's nitrogenous bases. And as you can see here, we have 4 different colors. That means we have 4 different nitrogenous bases that code for our genes. So all the stuff that basically makes up who we are basically inserted into these bases. They code for everything: your hair, your skin color, your height, though how big your hands are going to be, blah, 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 so on and so forth. So those 4 bases are called adenine, so represented by the letter A, thymine, represented by the letter T, cytosine, represented by the letter C, and guanine, represented by the letter G. So these bases have pairs, and it's due to the way that they're structured, in which I will show you here in a little bit. So A pairs to T, and G pairs to G, and C pairs to G. So this essentially will always be true. And we'll see later on in the unit what happens if they're not true. So what happens if they don't pair up with each other that way? So when you look at the structure, so base pairs basically connect both sides of our DNA. And like I said, A connects to T, G to C. What ends up happening here is that there's something called a hydrogen bond that sticks both strands of our DNA together. So these hydrogen bonds are very particular in that because these are base pairs, they have a certain number of hydrogen bonds. So adenine and thymine have 2, and guanine and cytosine have 3. So if we were to misplace these and swap out, let's say, for example, A with G, G and T cannot pair with each other just because of these bonds. And also these bonds are important because this is basically what holds our DNA molecule together. So recap here. So our DNA is double-stranded, right? So this double helix, double-stranded. So we know that it's a helix. And I'll talk a little bit about here in a moment about why it's called a helix. Why do we know it's double-stranded? Why do we know it looks like a spiral? So we have 4 nitrogenous bases. So thymine, cytosine, adenine, and guanine. So see how the structures here kind of differ. So when we pair them up, adenine and thymine are basically the perfect pair to pair up together, and then cytosine and guanine. So like I said, they will always pair up in that way. So as you can see, yellow and red are A and T. So apple tree is something to kind of remember it by. That's how I remember apple tree. And then cytosine and guanine would be like, would always pair up with each other. So the blue and the green. So you can basically represent them by different colors. So let's go on, move on. So one very important property of DNA is that it can replicate or make copies of itself. So DNA can always make copies of itself. That's why it's double-stranded. That's why we have two strands here. And later on, we'll talk about how DNA replicates itself. So when I look at my DNA strand, so typically you guys are going to see it in this format on paper. So my 5' starts, so this is where my DNA starts on this side from, and then I have my bases. So see how I paired them up: A and T, T and A, C and G, G and C, C and G, G and C, A and T, A and T, so on and so forth, right? So 5' to 3', and then I have my 3' to 5' here. So this is my DNA strand that is basically on paper here. So you'll basically see it like this. So you should know how they pair up, and you should be able to recognize how it looks like, at least by the end of this video. So moving back on to why we know that DNA is a double helix, so double-stranded. So when we look at scientists and basically science history, how do we know about DNA? How do we know it's double-stranded? And it's the way that certain scientists, so in 1953, Francis Crick and James Watson basically discovered the structure of DNA. So they ran this image of DNA under a microscope back in 1953, and they saw that it kind of looks like an X, and it has like this little ring forming around it. So they can see that these black things are the base pairs, and this thing that forms around it is the spiral that we talked about, the phosphate, the sugar phosphate backbone of our DNA, of the nucleotide. But so that's how we know that DNA is double-stranded. So something I did want to mention was that actually Francis Crick and James Watson did not really discover the structure of DNA, and rather so it was discovered by someone called Rosalind Franklin through the use of X-ray crystallography, where X-rays are beamed through the molecule to create shadow pictures of the molecule structure. So Franklin did most of the work at the time, but due to the circumstances back then, it was 1953, they didn't really recognize a lot of women's work. So her work was credited under the other scientists, Crick and Watson, without her knowledge. So they basically stole her work, and they were the ones to propose the double helix structure. That could explain how DNA could replicate itself during cell division. So those other two guys, Crick and Watson, won a Nobel Prize in 1962, whereas Franklin did not. But now we know that Franklin was our main scientist here, in which she did all the work to basically discover DNA. All right. So moving on. So what you should know, hopefully by the end of the video, is what is DNA? What is a nucleotide? What are the 4 nitrogenous bases? So remember, A, T, G, and C. A and T pair up. G and C pair up. So our bases in the DNA, what do? They code for our genes, the base pairs, and the structure of our DNA. So it's double helix, it's anti-parallel, has hydrogen bonds, and it goes from 5' to 3', and you flip it. All right, guys. So if you're confused about anything, you can always just rewind the video. I hope this helps. We'll see if this is something we can do in the future.